today's topic. And I think now you have got a basic idea about what are the stem cells, how the stem cells are look, how the lymphocytes are grown in the cultures, and how they can be amplified. So here, if you see this picture, I have already told you about the CCR5. Now when there is CD4 cells, they have along with CD4 receptor, they have the CCR5 receptor, which is the binding point to the HIV virus. That is the entry point. Now, if the concept is that if we remove this only, then there will be no, and here you see, without CCR5, or making a mutation. See, there are two ways. One is chop out CCR5 by CRISPR technology, or make the mutation. So any of this, then the virus will only remain in the front. It will not affect the CD4 cells, and your immune response will not go down. So this is here also I have shown you the mechanism of HIV virus infecting CD4. Here you see this is a virus which is in between. This is a CCR4 and CXCR4. These are chemokine receptors, and here it is entering. This is a RNA-based virus. Now this particular signaling can able to control because of using the anti-ART, that is antiretroviral drugs like Inio4 and various. There are 20 drugs which helps in interfering this. But what is happening is that this is a lifelong treatment which we want to terminate now. We want in such a situation, by using the stem cell concept, that the virus will not enter into the CD4 cells. So we have to make that particular cell so that this can prevent the HIV. So this is another basic mechanism I want to show here. You see, this is an unmodified cell. This is a transient knockdown. Suppose there is some patients who doesn't have the CCR4, then this patient can become a resistance, but some of them may develop suppression, loss of this, and they become susceptible. But when you chop out or knock down this CCR5, these various terminology like GFN, TELN, and CRISPR, you can mix the CCR genes with elimination of the CCR5, and the HIV resistance can be developed. And third concept is the stably integrating delivery of the various vector resistance to gene to this. And so these are the people are now trying to work on this concept, and mainly the clinicians are taking easier concept, which is the one which is they are taking in that they are taking directly the donor who has got CCR5 negative resistance gene, that is CCR5 32 delta. So these are the 2-3 studies they have used this thing, whereas some of the scientists, they are working on this concept and going further for this. So within I think 1 year time or 2 year time, I think many papers will come on this area, and mostly they will publish in Nature. Even you people can work, those who have good laboratory, they can work in this concept and really do the good work, and which will be publishable in very good journal like Nature, Science, or Lancet. So, but you must have the thorough work in this particular stem cell research as well as stem cell technology. So you will be technologically expert. So here also you see the CD4, this is a receptor entering. And here, this is a mutant donor cells, which are injecting to the patient. And when these cells are there, these cells doesn't allow to grow these cells, but these get multiply. Therefore, the virus remain away from these cells, and the cells become intact, and the immune response increases. So this is a general protocol which I have shown you, that the patient's HC hematopoietic stem cells has been taken. They are grown in culture and they're injected into the patient, and that way they see the follow of this patient for getting the proper remission of this HIV disease, and they have successfully done. Now this was the first actually stem cell transplantation has cured HIV infection in Berlin patient, and see how powerful that program was. The doctor want to take even patient along with this. This person is become also famous because he has cured from HIV. You know, not only doctor become famous, but his name is also coming everywhere. Patient Brown's work and this thing, So Dr. Jiro Hutter, who carried out a stem cell, it has been proven adds patients cured with stem cell from HIV resistant donor, you know. So here what they have done is John Hutter, who carried out a stem cell transplant on HIV infected man with leukemia in 2007 and cured HIV infection as a result of the treatment, which introduced stem cell which happened to be resistant to HIV infection. So the donor is HIV resistance, and that's why they have given it to this patient. The man received bone marrow from the donor who had natural resistance to HIV infection due to his genetical profiling, which leads to CCR5 cooperative being absent from his CD4 cells. Most variety of HIV uses CD5, CCR5 as its docking station, attaching to it in order to enter and infect CD4 cells. So people with these mutations are almost completely protected against infection. So this is a concept they have taken, and this they have really make a breakthrough and first report showing that stem cell therapy can cure. But here there again bone marrow transplantation. That point I am coming ahead also next, because how it is difficult. Now, this is one of the experimental model which is shown by the Dr. Dukes and Dr. McConney. Here he has clearly shown how he has done the knockout by this gene program technology. Now here you can use more easier technologies, CRISPR technology, and this is no CCR modification from this human cell, human stem cells, sorry, hematopoietic stem cells. Blood cells means these are the lymphocytes, and they have seen and they have injected into mice, and then they have seen the HPC and thymus mediated expansion of the peripheral CD4 cells. And here you see the low vironoma, you see there are more maintenance of CD4 cells. This particular experiment also done by Dr. Deshpande. I'm coming to that report in the next two slides. Okay, and here you see where there is a hyalinoma where you have injected the cells with CCR5 modification. So this is clearly indicating that chopping out the CCR5 genes may help in doing the curing the cancer. Now this is again a human trial, done clinical trials done by stem cell transplant specialist Dr. Fiona von Bessian, Dr. Hosu, and infectious with Dr. Marshall Ha from Weill Cornell Medical. He has also followed up the same way as the Dr. Hutter has done it. He has taken HIV positive human who received a blood stem cell transplant to treat acute myeloid leukemia, appear to have been cured of HIV. The stem cell from the umbilical cord blood contain a gene variant that makes them resistant to HIV infection. The woman has been free of HIV for 14 months since the treatment. This finding could point toward the cure of HIV for some patient. Now, one question you will ask, how they are assessing that they have cured the HIV? As already I have told you the testing of this. They are doing just CD4 testing. And when the CD4 counts are going up, that means the patient is in better state. This is a very important indication of this is a preliminary observations they monitoring, and the final things they can do the viral load by RT-PCR method. Again, this is the first woman treated with bone marrow transplantation cured by HIV. This is again two scientists, Yone and Dr. Deborah Persidis from Johns Hopkins Institute and UCLA. They have also made the similar experiments, and they have shown that their patient has better cure from the cancer. So there are almost 3 studies which have clearly shown that during the bone marrow transplantation of the cancer patient, they are treating the cancer, but when they are treating the cancer, they are taking the bone marrow from the patient who are having the CCR5 resistance gene, and that cures the cancer. Now, this is a second patient free of HIV after stem cell. This is a Dr. Ravindra Gupta. I think he is Indian, but his work is tremendous work he has done. He's an infectious disease physician at the University of Cambridge, UK. Picked up the donor who had 2 copies of mutation in the CCR5 gene that gives people resistance to HIV infection. Now here what he has done is little bit, everyone is making different strategy, and that is very important for the research. You cannot just go on one this. You can do the bone marrow transplantation, take another patient who has this and inject into that. This whole things exercise, you have to really reduce that. What he has done that he has seen the 2 copies of mutation in the CCR5 gene. Now, how we detect the CCR5 mutation? That is done by the sequencing, sequencing method. So slightly you may be now getting idea that Omicron, you need the sequencing, DNA sequencing. And there you can see the mutation. Mutation is nothing but a change in one of the nucleotides of that particular sequence. So they have taken the patient who has got a mutation, and the gene codes for the receptor which site on the surface of the whole blood cells involved in the body immunoresponse. Normally, the HIV binds to this receptor and attack the cells, but deletion of the CCR5 gene, that is the same concept which I already told you. About 1% of the people of European descendant have 2 copies of this mutation and are resistant to HIV infection. So see how Dr. Ravindra has made it more simpler in treating the HIV, but he has to further study it. And I think in 1 year time, I hope that a lot of things will come out with the positivity. So Gupta team described the results in the paper. This is published in Nature. I told you just now, if you do some of the work, see, now I don't have any laboratory or something, but anybody wants to collaborate my idea about this, and they have a good laboratory, especially mainly in Bombay, because I am in Bombay, I stay in Bombay. But if it is outside also nowadays, working on the online or going for some meetings, some just a visit, doesn't matter. So if anybody of the clinicians have the idea, or students have this, or institutions, I can definitely help them to do this work in actually. So this is really a good work of Dr. Gupta, and that's why I want to mention it here. And this is her work, Satya Dandekar, who is also seems to be Indian professor, but maybe she may be from the US citizen. So she has done work in US Davis. Now her work, her concept is really fantastic. See how the clinicians and scientists work together, get a better results. What she's observed is that all the HIV patients, their CD4 counts on gut means in the abdominal or some gut cavity are reducing. They are more, they are more susceptible to the HIV infection. And that they found out. And what she thought that if we can make mesenchymal stem cells, everyone knows it, they are very immunomodulators. So whether these cells, that's why I showed you my work in the beginning, you should know about the mesenchymal stem cell also. Whether injecting those cells in that particular region can elevate the CD4 cells, and she got a positive thing. So this is a whole thing. I will just read it so that you will definitely get a good, the groundbreaking studies by the team of UC Davis researchers has discovered a special type of stem cells that can reduce the amount of virus causing AIDS, boosting the body's antiviral immunity, and repairing and restoring the guts, lymphoid follicles damaged by the SIV virus and HIV virus. I'd say almost same, it works on mechanism. The equivalent to human HIV virus in non-human primates. She has worked this on not in the human, but she got the observation from the human, and she has worked on the human primate. Impaired immunofunctions in HIV infections and incomplete immunorecovery was obstacle for eradicating HIV, said Satya Dandekar, senior authors of this paper. Our objective was to develop a strategies to boost immunity against the virus and empower the host immune system to eradicate the virus. We thought to repair, regenerate, and restore the lymphocyte follicles that are damaged by the viral infection. So here she has used totally regenerative concept of the stem cell therapy in making the HIV patient cure. She has not done any clinical studies, but she has done in the primate studies. This person also has worked on the animals and shown that the promising result from Dr. Prashant Kumar Das. He is from University of Nebraska Medical College, 2009. Best work, promising early data from Prashant Das, 2009 suggests that durable HIV remission might be achieved with a genetically manipulation carried out in vivo. Studies in humanized mice using adeno-associated virus delivery, CRISPR targeting. See, he has used CRISPR technology, which is presently a hot topic because this is you got a Nobel Prize also, and it is very, very easy and useful technology for making the gene editing. HIV provironal DNA resulted in the reduction of replication competent virus in the some animal with without off-target effect. These preliminary studies reflected trend towards genetic modification as a new tool that with time will hopefully become both feasible and safe for the patient also, and it will be less costly and less cumbersome from making the bone marrow transplantations. So tool which we have used for the modification of CCR5 is the multiple tools exist. One is transcription activator like effector nucleus, TLN, fingerprint nucleus, ZMNS, cluster regulatory interface for palladium repeats, CRISPR. But nowadays, most of the people are using CRISPR technology, which is more easier. JFN has already been safely used to disrupt CCR5 ex vivo in circulating T cells by Tebas et al. 2004. Tebas et al. 2019 has reported single infusion of CD4 plus lymphocytes, modifying using CCR5 specific JNFN, which has delayed rebound in the individual who were CCR5 delta heterogeneous. So these studies are also been carried out in being work on this, and this will be really a breakthrough for in curing the HIV. So strategies to cure HIV is the antiretroviral therapy has become a increasing safe and effective, transforming HIV infection into the terrible chronic disease that is already going on, but it is a lifelong. However, given the morbidity and mortality associated with the bone marrow transplantation, such an approach could never become widely accessible as cure, because I have already told you that making bone marrow transplantation is one of the big procedure, to go for the patient as well as for their family as well as the doctors, so even they are cure HIV patient by doing the bone marrow transplantation and transplanting the donor cells, but this is a very complicated, so you need to have some alternative way. Why then has the announcement of the London patient generated so much? Of course, it has been generated a lot of interest because these are curing some disease, which is very complicated. CCR5 is one of the attractive targets for the first generation of genetic modified approaches. Nearly 1% of the people living in Northern Europe do not express CCR5. This is one of the very good important, and these individuals have normal life and generally do well. So this work also has to be done on these people, and you need to see how other people also be like this. So main concern about the stem cell therapy process in HIV infected individual, barrier to success is a delivery of the cells. Ex vivo description of the stem cell and mature T cells is feasible and likely safe, but short of full ablation followed by the autologous transplantation, only a small portion of the cells will be HIV resistance. Theoretically, if the patient engraftment occur and RT is interrupted, the virus will gradually deplete cell expressing CCR5 while sparing the lack of receptor. So only the lack of receptor, I have shown you the picture of this, only the CCR5 resistance cells will be grown. Over time, a fully resistant immunosystem comparable to the Berlin and London cases emerges. This may turn functional to generate de novo protective response against HIV. This approach will take time, necessary sustained period of viremia and associated risk, and may ultimately fail as virus that enter T cells via another co-receptor CXCR4 are selected. There are two receptors, but this is not been much in pictures. But of course, we have to see some of the patient whether they go in this cycle. For this approach to be scalable, CSCR will need to be edited in vivo with an as yet undefined proportion of potential target cells effectively modified before ART stop. So this is a main thing. So we'll come to the excitement in the infection disease scientific committee to cure is the enhancement of second individuals. This I already talked to you. That's why the HIV become ART-free viral remission. In similar case to the first cure, Berlin patient and HIV infected man in London underwent an allogenic bone marrow transplantation for treatment for the underlying cancer. The donor was homozygotes for deletion of CCR5. CCR5 is required co-receptor for host. The patient rebuilt immune system was naturally resistant to HIV infection. And in conclusion and future direction, we are now aim to develop a cure of HIV, mainly devoting to the shock and kill. Mainly shock and kill is the antiviral treatment. Block and lock, which is the present, which we are doing other stem cell therapy. And immunotherapy is a new branch where you can increase the immune response of your cells, or you can see how your mechanism, you can make the, you must have heard about the CAR T cells, So such type of innovations are already going on. So with the goal generating the remission. So there are a lot of avenues. In case of HIV, we can work on it because it is targeting to the CCR5 deletion or CCR5 mutations. So the clinical course of Berlin patients suggested that CCR5 destruction may be curative, and the London case in the consistent with the hypothesis confirming that by repressing CCR5 impression on the target cells may provide durable state-of-art ART-free viral remission. So this also can be done if some HIV patients are going under in bone marrow transplantation. This particular concept can be useful for them. With further refinement, this approach, suppression and protection, might prevent post-ART viral rebound and result in the safe, scalable state of durable viral remission for those living with HIV who cannot access or are not able to remain on RT. So this is a whole concept which I really explain you in the depth. If anybody has question or something,